An amiable welcome to everyone. This is your allied Lecdem channel to enhance your understanding. So, today's topic is about Leukopoiesis. So, if you guys missed the erythropoiesis lecture, you can check out with the previous video. Now, let's get into the topic, Leukopoiesis. The etymology shows that the word leuko refers to white cells and the word poiesis refers to production. So, leukopoiesis is one of the process that comes under the hematopoiesis, in which white blood cells, which is also called leukocytes, are formed in bone marrow that are located in bones in case of adults, and in case of mid-gestational fetus, it is located in hematopoietic organs like liver and spleen. Under normal conditions, that is in healthy individuals, there are 5 types of matured white blood cells or leukocytes that are found in peripheral blood. Among that 5 types of cells, 3 types of cells are called granulocytes as they contain granules in their cytoplasm. Depending on the nature of granules that are present in the cytoplasm, the cells are named as neutrophils, eosinophils, and basophils. The other 2 types of cells are called agranulocytes as they lack granules in their cytoplasm, and they are named as monocytes and lymphocytes. Among these 5 types of cells, granulocytes, that is neutrophils, eosinophils, and basophils, along with monocytes, are formed in the bone marrow from a pluripotent stem cell, a cell which is responsible for initiating the development of each and every cells of our body. So it takes place by 3 phases, that is differentiation, proliferation, and maturation. Then the maturation process of lymphocytes is a very complex method, and it takes place in the reticuloendothelial system. The blood granulocytes along with monocytes are controlled by a feedback system which is similar to that of RBC'S, that is as like in erythropoietic system. The colonies of particular white cell line is produced and stimulated by several factors that include colony stimulating factors, which is also called leukopoietins. So here there are certain criteria for identifying the cells that include size of the cell, ratio of size of nucleus to that of the cytoplasm, appearance of nuclear membrane, presence of nucleoli, staining reaction of the cytoplasm, presence of cytoplasmic granules, and the type, size, and distribution of the granules in the cytoplasm. Development of granulocyte series, which is also called myeloid series or granulocytopoiesis. When blood and bone marrow aspirated cells are stained with Romanowski stain, the following cells can be shown. Here comes the diagrammatic representation of granulocytopoiesis. The developmental stage are same for all granulocytes. So here, from the pluripotent stem cell, MYELOBLAST is developed. Then promyelocyte, myelocyte, metamyelocyte, stab cell, which is also called band cell, and finally the granulocyte, pluripotent stem cell. The size of the cell is usually 25 to 40 microns and it have an irregular margin with blunt protoplasmic projections. Its nucleus is round and large and its nuclear chromatin is finely reticulated and arranged with purple-red stain. Nucleus to cytoplasm ratio is about 1:1. NUCLEOLI is 1:4 and it appears pale blue, well defined and it is irregular in shape. Cytoplasm. It appears blue and non-granular, that is, no granules are present at this stage and it is less deeply stained near the nucleus. MYELOBLAST. The size of the cell ranges from 10 to 18 microns in diameter and it is ovoid or oval shaped. Then nucleus. The displayed blood picture shows how the nucleus occupies most of the cell and it is round or slightly ovoid in shape. Nucleus to cytoplasm ratio is approximately 6:1. Its nuclear membrane is smooth and thin, while the nuclear chromatin is finely reticulated, evenly distributed and stained purple colored. NUCLEOLI present here is 2 or more and it is distinct, that is well defined and ovoid in shape and stains pale blue. Then cytoplasm. As the nucleus occupies most of the cell, the cytoplasm is very thin. As it is basophilic, it appears blue color at the upper edge of the cell and less basophilic when compared to the pronormoblast. Next stage is promyelocyte. The cell size ranges from 12 to 20 microns and it is round or oval shaped. As the blood picture shows, the nucleus is ovoid, large and also with slightly coarse clumping, that is, the clump that forms is little rough and the nuclear chromatin stains light purple. The nucleus to cytoplasm ratio is approximately 5:1. Here, the nucleoli is 2 or more and it is less distinct, that is, it is not that much well defined than the blast cell, that is immature cell, and it is ovoid and stains pale blue. Cytoplasm appears light purple and basophilic with few relatively large dark blue granules starts to develop at this stage, overlapping the nucleus. Myelocyte. The size of the cell ranges from 12 to 18 microns and it is round or oval shaped. Nucleus is round or oval shaped and it is indistinct with more coarse clumping of purple chromatin as present in the blood picture. The nucleus to cytoplasm ratio is approximately 2:1 and here the nucleoli is usually absent. Cytoplasm appears bluish pink with easily undifferentiating acidophilic granules that are primary granules and it is present in more numbers and they are smaller in size when compared to promyelocyte. They start differentiating into secondary granules such as chunky orange red color granules in case of eosinophils, blue black granules in case of basophils, or lilac, that is a Eurasian share, which is pink in color in case of neutrophils. Metamyelocyte stage. The size of the cell ranges from 10 to 18 microns and it is round or oval shaped, and it is slightly smaller than the myelocyte, and it can also be differentiated from the myelocyte by the shape of the nucleus. So here, as the blood picture shows, the nucleus is indented, that is oval shaped or kidney shaped. The nucleus to cytoplasm ratio is approximately 1.5:1. The nuclear chromatin present here is dark, purplish and coarse. Then cytoplasm is abundant and stained pinkish blue color and filled with numerous small granules. Stab cell or band cell. It is also called juvenile granulocyte. The size of the cell is from 10 to 16 microns and it is round or oval shaped. The shape of the nucleus is sausage, which is an cylindrical shaped food item, or horseshoe shaped as present in the blood picture, and also with some occasional areas of constriction. The nucleus to cytoplasm ratio is approximately 1:2, and the nuclear chromatin appears coarse and deep purple blue. Here, the nucleoli is usually absent and cytoplasm is large in amount, and it is pale blue or pink with fine lilac color granules in case of neutrophils, large chunky orange granules in case of eosinophils, or coarse blue black granules in case of basophils. Polymorphonuclear granulocytes. It is the final and matured stage of granulocytes. So here the cell's size ranges from 10 to 15 microns, and the nucleus that are present in younger cell is purple in color, and it consists of two lobes. Those are separated by a strand of chromatin. In case of older cells, they have 3 or more lobes which are separated by a very thin chromatin strands. The nucleus to cytoplasm ratio is approximately 3:1 and nucleoli is absent. Cytoplasm appears light pink to blue. As this is the matured stage of granulocytes, the granules of neutrophils appears small violet pink, and the eosinophil has large bright orange granules, and the basophil has large coarse blue black granules. So these granules completely fill the cytoplasm and obscure the nucleus. That is, the nucleus is not well expressed. Note, as the development stage has 3 phases, here the stages from 1 to 4, that is from pluripotent stem cell to myelocyte, it is the proliferation phase of development, while stages from 5 to 7, that is from metamyelocyte to polymorphonuclear granulocytes, it is the differentiation and maturation phase. Development of the lymphocyte series. Lymphoblast. It is the earliest cell of lymphocyte series, and it is present in bone marrow on some occasions but in small numbers. It is difficult to differentiate it from myeloblast cells. The size of the cell is 10 to 18 microns. Then the nucleus occupies most of the cell and it is round or oval shaped. The chromatin present here is dark purple and aggregates along the nuclear membrane. It is coarser than myeloblast. The nucleus to cytoplasm ratio is about 6:1. The nucleoli present here is about 1 to 2 and indistinct with light blue color. Cytoplasm appears deep blue with no granules, and most of the cells show paler perinuclear area, that is, a surrounding area of the nuclear looks paler than the other area. Here, the blood picture shows the lymphoblast cell, which is denoted as LB, and the lymphocyte, which is denoted as L. Prolymphocyte. The size of the cell ranges from 9 to 17 microns. The nucleus present here is ovoid or oval in shape and occasionally indented border. That is, the border is kidney or oval shaped. Chromatin is coarser, darker and more aggregated than in the lymphoblast. The nucleus to cytoplasm ratio is about 4.5:1. Nucleoli is 1 and appears blue in color. As the blood picture shows, the cytoplasm is more than in the lymphoblast with light to dark blue color and occasionally with eosinophilic granules. Lymphocytes. In humans, bone marrow and thymus forms primary lymphopoietic organs where the lymphoid stem cells undergo spontaneous division and it is independent of antigen stimulation. There are two types of lymphocytes, B cell and T cell. The lymphocytes also classified as small lymphocyte and large lymphocyte. The size of large lymphocyte ranges from 8 to 16 microns and small lymphocytes ranges from 7 to 9 microns. Nucleus is usually round in shape and frequently eccentrically located. That is, it is located far away from its place. Nuclear chromatin is dark purple, blue, very coarse and clumped. The nucleus to cytoplasm ratio for large lymphocytes is 1.5:1 and 1.25:1 for small lymphocytes. Nucleoli are usually absent. The cytoplasm appears light blue, and as the blood picture shows, the cytoplasm varies for small lymphocyte and large lymphocyte. In small lymphocyte, it is present as a ring around the nucleus, and in large lymphocyte, it is relatively abundant. And in large lymphocyte, occasional eosinophilic granules are seen. Here comes the some type of lymphocytes like natural killer cell, CD8 cytotoxic T cell, B cell, regulatory T cell and CD4 helper T cell. Development of monocyte series. This shows the development and differentiation and their respective compartment or organs in which they are taking place. The myeloid progenitor cell is developed into monoblast, then promonocyte and monocyte, and this development takes place in bone marrow. Then the monocyte enters the blood vessel. Then it is divided into dendritic cell and macrophage in tissues. Monoblast. The size of this monoblast ranges from 12 to 20 microns and its nucleus appears large, round or ovoid as in the blood picture, and the nuclear chromatin appears light purple, pink and reticular, and the nuclear membrane is very fine with a distinct and defined manner. The nucleus to cytoplasm ratio is about 2:1, and here the nucleoli is present about 1:2, and cytoplasm appears light blue without any granules. Promonocyte. It is the intermediate stage between monoblast and matured monocyte. The size ranges from 12 to 18 microns, and as in blood picture, the nucleus is large lobulated, that is, it has many lobes and kidney shaped with fine light purple thread-like chromatin. The nucleus to cytoplasm ratio is approximately 2.5:1 and nucleoli is 0:1. Cytoplasm appears grey-blue with large and small lilac acidophilic dust-like granules. Next is monocyte. This is the largest cell of normally occurring peripheral blood cells. Here the size is about 12 to 16 microns and the nucleus is oval, kidney or horseshoe shaped. Fine lacy delicate chromatin stained light purple pink, and the nucleus to cytoplasm ratio is approximately 2.5:1, and at this stage the nucleoli is completely absent and the cytoplasm is abundant, and it appears slate grey color with many fine lilac colored granules and vacuole. Development of plasmocyte series. Plasmocyte is a cell that are formed by transformation of antibody producing lymphocyte, especially B lymphocyte. When stimulated by an antigen, these B lymphocyte transforms themselves into a plasmoblast, which further proliferates and matures into a plasmocyte, which is also called a plasma cell. So, these plasma cells are responsible for producing antibodies which are specifically directed against the stimulating antigen. Plasma cell development. It is the diagrammatic representation of development of plasmocyte. From white blood cell, the lymphocyte arises and classified as B cell, T cell and natural killer cell. And here the B cell especially develops into a plasma cell which produce antibodies against the antigen. Plasmoblast. It is normally not present in blood but rarely present in normal bone marrow. The size of the cell ranges from 14 to 24 microns and its nucleus is eccentrically placed and looks like somewhat egg-shaped and present usually in the narrower pole of the cell. The chromatin is reticulated, that is finely arranged, and appears purplish and coarse. The nucleus to cytoplasm ratio is about 1:2:1. Nucleoli is 1:3 and large. Cytoplasm is relatively abundant, deeply basophilic with occasional areas of light staining. Proplasmocyte. Its size ranges from 14 to 22 microns and nucleoli present here is large and it is 1 to 2 in number. Cytoplasm is abundant, light blue with lighter perinuclear halo. Plasmocyte, a plasma cell. It is egg-shaped, that is narrower at one end and looks broader at the other end. Its size ranges from 8 to 18 microns and its nucleus is ovoid or oval shaped and it is eccentrically located, usually in the narrow end of the cell. The chromatin appears purplish and extremely coarse and gives a cartwheel appearance. The nucleus to cytoplasm ratio is approximately 1:2 and here the nucleoli is absent. The cytoplasm appears deep blue with pale perinuclear halo near the one side of the nucleus. Vacuoles may be present near the cell border. It is the blood picture of plasmocytic series, that is plasmoblast, proplasmocyte and plasma cell. Functions of leukocytes. Firstly, neutrophil. It plays major role in phagocytosis, febrile response and reaction of inflammation. Eosinophils play their role in allergic reaction, immunity and in parasitic infection. Basophils release heparin, involve in phagocytosis and play their role in preventing spread of allergic inflammation process. Lymphocytes, as it contains two types of cells, B cells which are responsible for development of humoral immunity and T cells, it is responsible for development of cellular immunity which is also called cell mediated immunity. Monocytes play their role in defense mechanism, that is when monocytes combines with neutrophils involve in phagocytosis and when it combines with lymphocytes it kills tumor cells. Regulation of leukopoiesis. It is the mechanism or process to control the production and release of leukocytes. The rate of production and release of leukocytes is increased by bacterial toxins during tissue injury and inflammation. The substances that stimulate and inhibit the process are complex. Ways to maintain your WBC's count. It's better to avoid sugar and sweet stuff as it prevents or lowers the strength of white blood cells. Also, it is important to eliminate unhealthy fats like polyunsaturated fats which is present in vegetable oils such as corn, safflower and sunflower oil, which inhibits the healthy immune system. Stay hydrated by drinking plenty of water to boost your immune system as well as to flush out all the toxins. And more importantly, avoid stress and try to relax. As stress is rightly called as silent killer, it can lead to a lowered immune system. Food that increases your WBCs. This includes citrus fruits like lemon, orange, then ginger, garlic, green tea and poultry. Thank you.